Okay, so neurotransmitters, where do these neurotransmitters come from? Well, it turns out evolution is quite conservative, and so very commonly available substances in the body are actually utilized as neurotransmitters. They're generally tweaked or changed or altered in structure to some extent from their original form, which will require proteins that are known as enzymes to actually help to synthesize these neurotransmitters from these widely available substances. So think of some widely available substances in the brain, in the body. These include amino acids, right, which are the building blocks of proteins, of all these protein machines that we've been discussing. There are also vitamins, or there are also byproducts of cellular metabolism, etc. There are short proteins that may be utilized as neurotransmitters. So neurotransmitters, one of the key facts to know about them is that they are manufactured from pretty widely available substances. And the protein machine that's necessary to actually manufacture these neurotransmitters from these widely available substances are enzymes, and they're called synthesizing enzymes. So let's look at what's happening here. This is a sort of standard axon terminal, right? Presynaptic membrane here. You can tell again by the vesicles, by the vesicular transporters, right? By the calcium channels. And here we've got what I refer to as the raw materials. These again are going to be, let's say, a byproduct of the metabolism of fat. Everybody has fat in the body, and all your cell membranes, right, are phospholipids. So there'll be substances that are associated with the metabolism of fat. It will be everywhere. Amino acids, maybe, or something would be another, commonly available raw material, because you have to build proteins to build all these structures. So this enzyme, which is a protein that your cell will manufacture, your neuron will manufacture, if this neuron is going to release this particular neurotransmitter, it'll actually express the gene, right, to build this enzyme that will synthesize this neurotransmitter, build it using these raw materials. So in neurons that are going to release, neurotransmitter X, this one right here, right, you're going to be, this particular neuron's going to generate this protein from the gene, and this protein is going to grab onto these raw materials and, put them together and synthesize this enzyme. Think about what else this neuron will actually do. If this neuron is going to release neurotransmitter X, it will not only synthesize neurotransmitter X, right, from these raw materials, it will not only express this synthesizing enzyme, it will also express a specific transporter. Remember, the transporters are proteins that grab something from one side and force them over to another side against a force, right? In this case, you're going to be forcing neurotransmitter against the concentration gradient force to pack that neurotransmitter inside, the vesicles at the axon terminal. So a neuron that's going to release, neurotransmitter X is going to express the synthesizing enzyme, which grabs these raw materials and builds neurotransmitter X. It's going to express the transporter, right, the vesicular transporter that's going to pack neurotransmitter X inside the vesicle. And then, because neurotransmitters are powerful, potent chemicals, right? They allow for the postsynaptic membrane to, receive a message, right, via receptors, and then respond in some fashion that can be quite dramatic, involving, perhaps rebuilding of that connection, right? Expressing new proteins, changing gene expression, protein synthesis in the next neuron, a lot of major changes. These are potent chemicals that will need to be broken down. You want these the influence of these neurotransmitters to be short-lived, right? You want it to send a message. Upon arrival of the action potential, there's release of the neurotransmitter and there's stimulation, right, of this postsynaptic receptor. But then the message ends, right? So in addition to synthesizing enzymes, generally there are what we refer to as degradative enzymes. These are enzymes that will break down neurotransmitters. They'll grab onto the neurotransmitter either out here in the cleft or perhaps inside the axon terminal itself, and they will take that neurotransmitter and they will split it apart so it can no longer influence receptors on the postsynaptic cell.